Okay, good afternoon, everybody, and welcome to Dear Editor, our conversation series from the Research Medical Library on scientific writing and publishing. And as always, after today's session, we are recording today's session. And after we're done today, I will share a link to the recording, and we will have a handout available summarizing all of the things, the information that's going to be presented this afternoon. Leading today's Dear Editor is Amy Nanetto and Joe Munch, two of my editing colleagues in the library. So with that, I'm going to turn it over to them.
Okay, before we get too deep into it, just show of hands or someone want to unmute and tell us, does any or everyone have experience with using iThenticate here?
I use it occasionally.
Okay. Anyone else?
I have not used it.
Okay, so we're going to go through, sorry? Okay, so we're going to go through some of the kind of the pitfalls and why you really shouldn't be too worried when you get your iThenticate report back and it shows that you've got an enormous similarity index. And so, Amy, I'm going to go ahead and share my screen.
Will do.
Okay, and you should be able to see a mock-up of a manuscript. And so let's see if I can make sure I can see everything here. So the first thing I think a lot of people are concerned about is that similarity index, and here it's 46%. And journals will use iThenticate to kind of vet their submissions to make sure nothing's overlapping substantially. And I think the cutoff that they use is 25%, but that's not a hard and fast rule. And any journal editor worth their salt will probably take a nuanced approach to interpreting these reports. And so in this case, I've created a mock manuscript. I grabbed something off of PLOS One from, I think it was like 2006. I messed around with it and switched out terms and jumbled up the text so that most of it would not be just a sure repetition of the original article. So this is just for demonstration purposes. And here's some of the issues that we kind of run across when we're looking at an iThenticate report. So you're concerned about potentially repeating previously published text. You upload your manuscript and you get your report back. It's kind of summarized in this screen here. And you, to your horror, you see that nearly 50% of your paper has been copied. And that kind of sets the heart rate going, unless you know that this is not gospel, so to speak, because there's so many things that can contribute to this similarity index. And so a few of those, if we scroll through here, the title was retained and the authors, because I didn't mess with those for the mock manuscript. But for example, if you have done an abstract for the study that you're publishing now, and the abstract found its way into a proceedings issue, it's probably already online someplace. And a repetition of the abstract in this case is not problematic. So this is something that you still retain the copyright to. It's still part of your study. And so this is not something that would really contribute to that similarity index. Another thing is, I'm going to skip way down to the bibliography right now, but this should come as a surprise to no one, is that bibliographies, reference sections, have a great deal of repetition with previously published manuscripts, because so many manuscripts share citations. And in iThenticate, you can actually eliminate this biography or bibliography by filtering out in the settings and filters. And so if you exclude that bibliography, iThenticate recalculates that similarity index, and it's already down to 33%, down from 46. Let's talk about a few other things that we're not really concerned about when we're looking at an iThenticate report. So as you're scrolling through here, you can see things like these very short phrases. And I think you'll agree with me to say that these are not problematic, because for example, into any cell type from all three germ layers is too small a snippet of text to be worried about. It doesn't show you can't really track it alongside any previously published text. Now, if you take that in contrast to this part in red here, you can see that that's 2 or 3 sentences where if you were to hold up the original text with your own, you'd be able to track along. And that I would say is problematic and needs to be rephrased or revised or paraphrased. Some other things that aren't really problematic, things like specific terms. Lineage restricted stem cells with endothelial potential is an actual thing, and this is what the things are called. And IThenticate flags it, but it's not problematic because that's what the thing is called. Now, if this term appeared in the middle of a block of text, that would be concerning, but alone, not so much. And then there's other similar things like there's sentences so common that they've become almost cliche, like p-values of less than 0.05 were considered statistically significant. In virtually every scientific manuscript, you see some version of that sentence to the point where it's endemic. There's no way to avoid phrasing it that way. And so that would be a phrase that isn't problematic. And as I'm going through here, Amy, do you see any other potential issues or things that you might stop and look at but aren't really problematic?
No, I mean, just I think you're about to get to the things in the methods, so that's, I guess, the next place I would start to examine things.
Right. And along those lines, as we're going through IThenticate reports, and we know this because as a service to our authors and editing services, we'll run each and every manuscript we receive through IThenticate. People who submit their manuscripts have the option to opt out, but as a service to make sure that that people haven't inadvertently included previously published text in their manuscripts, we'll run the report. We'll run the IThenticate report. And so before we even lay hands on the manuscript to edit it, we'll take a look at the report to see if there's any outstanding issues that need to be resolved first. And one of the things that we generally agree on is that whereas some repetition in certain parts of the manuscript is acceptable, we'll talk about that in a little bit, repetition in other areas, not so much. So repetition in the introduction section is, I think we can say it's something that we should not see. But if we come down to the methods section, and this is another just kind of a function of science being so precise, I think, and some analyses being so complex, that there's not many different ways to phrase things. And so here you can see that a whole bunch of text for this RT-PCR analysis is flagged as appearing previously. And this isn't the best example because this is a fairly straightforward approach. When I see the great repetition of a method section, I'm usually seeing it in terms of a single cell RNA sequencing analysis where authors are talking about creating libraries and merging files and doing all these things are above my pay grade. But the point here is to say that some repetition in the methods isn't necessarily a bad thing as long as you do it, following proper guidelines. And that's something that Amy will talk about here shortly. But before we move to that, does anybody have any questions about IThenticate reports? I realize this is a kind of a thumbnail sketch of it, and I'm being very distracted by the sun is just now coming through my window, so I'm getting the glare. But any questions about IThenticate reports?
So, Joe, I have a question.
Yeah, Larissa.
Do you have a sense, I mean, we've talked about this before, but do you have a sense of how harsh publishers are about these reports? Or do you ever hear anything about that?
I haven't, I don't know, maybe Amy has a different take. I haven't heard anything. What I have seen is that publishers will run their manuscripts through IThenticate or a similar service, to see if there's any previously published text. And usually they mark a threshold of, and usually I see like 25%. What I don't know, but what I kind of presume just based on my own experience and how, as an experienced user of iThenticate, I would approach a manuscript that shows a similarity index of 50. Before I just dismiss it out of hand, I'm going to look through and make sure things like the bibliography have been excluded. I'm going to make sure that it's not just a hodgepodge of things that in fact are not very problematic. And I think that you can, when you get into iThenticate, you can go to the filters and setting, and you can exclude sources that are less than X number of words. So maybe the journal says, well, if a source doesn't have 10 words, it's not worth looking at. Or they'll exclude matches that are less than 10 words. And these are the criteria they use before they decide on that 25% similarity index. And so if you decide, if a publisher decides that, well, we're going to use these specific criteria, and I think that any publisher that understands iThenticate would, if they're not going through it page by page like we do, they're probably applying some criteria to make sure they're not getting just a hodgepodge of just snippets. Because one thing that iThenticate is good at is identifying previously published text. What it's not good at is saying whether the text is actually problematic. It's not good at saying whether the text, or whether rather the sources it identifies, are actually the original sources. And here's just a good case. Amy and I actually went through this earlier today just to kind of talk about this mock-up. And this morning when we went through it, when we ran the report, this journals.plos.org, that was actually like number 2 or 3 in terms of which the amount of repeated text it has. And that would be the original source for this article. That's where I got the article. But it identified some other random website as also having that text. And so it will flag the text that's been repeated, but for whatever reason, it won't always identify the original source. And so some cautionaries about that. And sometimes that original source is in itself not problematic, because it might be from a manuscript that the authors wrote, and they're now going to reuse some of their text in their new manuscript. And that's something that Amy's going to talk about right now. So I'll stop sharing.
And I will go back to sharing. While I'm doing that, yeah, I would just say that the journals, I think, pretty universally understand that iThenticate reports require some human interpretation. I think of them as kind of like lab tests. There's potential for false positives, and so it does require a human with some expertise to look at them. My understanding is that they do sometimes set cutoff points for those percentages of overlap, but are not particularly transparent about exactly what those are. And so sometimes it's a little bit difficult, that they don't want to necessarily reveal that information so that people don't just, adjust a word or two here to get under that percentage. But really, you do have to look at the report kind of in a holistic way and try to understand exactly what constitutes problematic overlap, which can actually vary depending on the purpose of that overlap or where it's located in the paper. So I'm going to talk about what used to be known as self-plagiarism, but is now more commonly called text recycling. And that's because calling it self-plagiarism, that is reusing your own previously published text, implies that there's something wrong with this, that it's equivalent to or similar to plagiarizing someone else's text, which is sort of thought of as a kind of intellectual theft, but you can't really steal something from yourself. So the idea of reframing it as text recycling is to identify appropriate places where reusing text might be okay. And one such place is in method sections. And this is just because, many kinds of research use the same methods. Research programs, may develop a method early in a project and then use that method throughout subsequent papers. And that gives a kind of unity to the entire research program. There are standard methods that, are kind of routine in labs, and it doesn't require a great deal of creativity to describe them. So here's an example of another an iThenticate report on a paper with substantial methods text that was recycled. So you can see here that, excuse me, I'm all hoarse today. We see a 61% overlap percentage, which is probably enough to set anyone who's looking at this is going to say, okay, we need to figure out what's happening here. And you can see that the large chunk of overlapping text appears in the methods section. This is just one page of it, but there are several sections in this method sections of this paper that are overlapping. And then you can see that the source of the overlap is identified by iThenticate here. And if you expand this, you can see that these are the same authors, Del Santo, Rice, and Carrier, and that the title here is actually, you can't see the whole thing on my slide, but the titles are very similar. So they're clearly related projects. They're related papers that used the same methods. So is this okay? Authors, since iThenticate became available, I think publishers and journals and authors have started to recognize that this is pretty common. And so they've started to develop guidelines surrounding what is appropriate text recycling. So Biomed Central, the publisher, and COPE, which is the Committee on Publication Ethics, publishing industry-wide group, have described text recycling as the use of similar or identical phrases in the methods where there are limited ways to describe a method. And they say it's not unusual and perhaps unavoidable. When you're using a technique that you've described before, when you're using a technique that is common between a number of papers, like we saw in that example I just showed, it's actually helpful to use the same words. It's actually helpful to not try to rearrange the text just to avoid iThenticate. That can introduce inaccuracy and make convoluted sentences. So BMC and COPE advise that editors use your discretion, use knowledge of the field to decide how much text overlap is accepted. And the key here, as with almost everything in scientific publishing, is to be transparent. So authors should alert journals that there is some recycled text, state that these methods have been used elsewhere or described elsewhere, and then cite them appropriately. The Text Recycling Research Project, or TRRP, is a multi-institutional project funded by the National Science Foundation. And its goal originally was to simply assess what practices people were engaging in with regard to text recycling. What was actually happening? What are scientists actually doing in terms of recycling text? And they then tried to use the information that they found to create some kind of guidelines or best practices for the ethical and legal recycling of text. So they defined text recycling a little more narrowly than the previous two organizations. The idea is that the material is identical or substantively equivalent to a source material. It is not a quotation, so we're not like pointing out like that we're bringing it into the text. It's just simply recycled. And the two documents, the source and the document containing the recycling, need to have at least one author in common. And so the principles that they're devising include that the recycled text should be accurate and appropriate. So just recycling a method that you used for a Western blot in an unrelated paper might not be appropriate. But using one that was part of this same larger research program might be more appropriate. Certainly, again, like Joe said, in the introduction or discussion sections, recycled text would, in the vast majority of cases, not be appropriate. Again, the corresponding author, they say, of the previously published article should grant permission. So there should be, you can't recycle text without, at least the corresponding author knowing about it. And the cover letter should include disclosure of the recycling. So again, transparency is the key here. So how do you comply with those guidelines about text recycling? The American Society for Microbiology says it's okay in the methods section, just cite it appropriately. The journal Anesthesiology permits text recycling only in the methods section and only to describe standard methods or standard protocols, and it must be limited. You can't just recycle an entire method section. The citation must be provided, and it must be obviously at least one of the author's own prior publications. So how do you disclose text recycling? So different journals might want it done a little bit differently. Some say in the cover letter, some, as you saw, say that citation is enough. In the example that we saw, actually, there was at the beginning of the methods section, this disclosure of the reason for the recycling. That is, they were using the same data set as several other studies. And they also state openly that they follow the guidance of the text recycling research project. So different journals have, slightly different standards and slightly different places that they might want you to disclose recycling and how to disclose it, maybe in the disclosure section, maybe in the methods, maybe only in the cover letter, always through citation. So you can work with the journal's editors about how to properly disclose. So Joe, anything I missed on text recycling?
No, I don't think so. As Amy said, the practices for it differ from journal to journal, but the common denominator is that this is an author's own previously published text, or at least previously published text that appeared in a paper that author was on. So this isn't taking a methods from a different group, sticking it in your own paper, even with and asking the corresponding author for permission, all that. No, this is that group's work. And I think that's important to remember.
Yeah, this is different from citing, the manufacturer's protocol, or different from citing a method previously published by another group. This is when it's your work in a project that's sort of continuous with the one that you're publishing. Anybody have any questions about reading these reports or understanding how you can appropriately reuse your own material?
One of the things I was thinking about earlier when we were looking at the iThenticate report with the overlapping abstract from a conference proceeding or something, if you're ever worried about that, I mean, you can always put a footnote on your title page that says, "Oh, this research was previously presented at such and such a conference," and then it'll be really obvious. Oh, yeah, that's a journal editor or somebody reviewing an iThenticate report would very much expect to see that overlap if you said that.
Yeah, and along those lines, I'm sorry, go ahead.
I was going to say the same thing. If you have published the manuscript as a preprint, Usually the
Right.
first hit and IThenticate will be bioarchive or something like that. And then you just, you can just exclude that source, and then, you know, journal editors will figure that out. But it's always a good idea to disclose it at the outset anyway, just so that, you know, they have a heads up that this is going to come up pretty high in IThenticate.
Okay, well, I think we can wrap, and we'll stop recording. But if anybody has a question that they want to ask us without us saving it for posterity, I mean, we'll hang around for a little bit.
I have a
Always happy to talk about these. Okay, sorry.
It's not totally directed at IThenticate, but there are a lot of times I have to assist my faculty member with their manuscripts. And whenever they have the manuscript reviewed by the library, first thing I do is when they ask me to submit it is automatically go and check to see if it's been edited. And if it hasn't, that's when I kind of go a little bit more into detail. Is there a checklist or something provided or that you have that will kind of give me a little bit more guidance where I don't have to just rack my brain a lot of times if it's not reviewed by the library?
Like of things that you can do to look for and IThenticate?
Not just in IThenticate,
Oh,
just
okay.
in submission in general, from using IThenticate to everything else that's required,
That
so
needs to
that
be
I don't
done.
miss a step.
I would say probably start with the journal instructions. Those are the kind of key pieces of things that you will need. Like I usually look at them and I'm like, okay, we have that, you know, and kind of make sure that all the information that they need is there. Make sure that everything's in the format that they want. And that can save you some work because some journals really care about formatting of articles at submission and some really don't. They're just like, just send it so we can, in a way we can read it.
Yeah, I use
And
that.
yeah.
I use that, but like sometimes I may forget to check and see if it's pertaining to a specific core grant or whatever. Right. I caught myself at the end before I submit the manuscript, and I went back and questioned my faculty member about it, and they said, "Oh yes, you're right. We do need to add that." I just don't want to miss anything.
Well,
we
that's
have a, yeah, sorry.
I was just going to say, you're talking about the overall submission process. Like what boxes you need to check before you actually hit the submit, correct?
Correct.
Yeah.
So I know
And
I
that's
use IThenticate, and then I'll just go through different steps once I read the instructions. But before the final submission, I just want to make sure that all my bases are covered. From
right.
using
And we
IThenticate to checking all the boxes in addition to what the journal may have for their instructions.
right. So not just what the journal says specifically, but in general, what you have to check for, you know, figures and tables and format and everything. And we don't have at the moment a specific checklist covering submission, but I think we should take a note of that, Erica and Amy, and I think that's something we might look into, having a good summary document for
Exactly.
pre-submission checks.
Because
That's a good idea. It's a guideline.
yeah, 'cause sometimes my faculty member, if they have time to get their manuscript submitted by their deadline, then they will use the library. But if not, then they'll send it to me and I'll have to go through and I'm being rushed and I just kind of get anxious and I don't want to have it returned for something that I failed to do.
Understandable.
Mhm.
Yeah, that would be something I think helpful for us to draw up. You know,
Mhm.
the checklists that we use internally are very, 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 very long and probably wouldn't be of much help when you don't have a lot of time. But there are definitely some essential items that we could pick out and put together.
That would be a great help. Thank you.
Great question. I see that Joe has put a copy of the handout into the chat if you are wanting to get started on that right now. I will also, of course, email it out to everybody who attended this morning as well. And as I said earlier, we'll have a recording available. And if you have additional questions, you get off of this call and think, oh, I should have asked such and such. I would also encourage you to join the Dear Editor team, which is also linked to on the handout. And you can always ask questions there. And of course, you're always at any time welcome to contact the library by email, chat, all the various ways that you can contact us, and we're happy to answer any of your questions. All right, I will stop the recording here.